Now we start protein synthesis, very important topic for question purpose. In protein synthesis, yes, before starting protein synthesis, you must know there is activation of amino acid. Activation of amino acid. What does it mean? Means binding of amino acid to transport RNA. Binding of amino acid to transport RNA. I have already told you there are 20 amino acid, 31 transport RNA. Means each and every transport RNA, yes, or each and every amino acid is having its own transport RNA. Each and every amino acid is having own transport RNA. 20 amino acids are there. Each amino acid is having one or its own transport RNA. Yes, understood. But there are 31, so few RNA. So each and every amino acid is having own transport RNA. We found that each amino acid has its own transport RNA, but few amino acids are rich. They are having more than one car. Transport means car. Each amino acid has its own car. Suppose this is transport RNA coding for serine. Serine will bind to it. This binding is done by amino acid transport RNA synthetase. In this synthetase use ATP. ATP is going to MP. When ATP is going to MP, two high energy phosphates are used equivalent to 2 ATP. So the question is asked, activation of amino acid, how many ATP used? 2 ATP used. Now suppose this is transport RNA for serine. Who will bind to this transport RNA? Serine. And who will check this is correctly matched or not? This amino acid transport RNA synthetase has proofreading. Question. Amino acid transport RNA synthetase has proofreading. Means you can simply remember transport RNA is car, serine is owner of the car, this is parking boy who takes energy, ATP currency of energy, yes, who takes money, yes, it is the responsibility of this parking boy to join amino acid correctly. Suppose this is transport RNA for serine. Can you tell me one answer? You recognize transport RNA by codon or anticodon? How you recognize that this transport RNA is for serine? How you recognize transport RNA that it is for serine? You recognize transport RNA, this transport RNA for serine with the help of anticodon. Anticodon. Understood? So this is car plate, number plate. This is number plate. So this transport RNA is for serine. Who will bind to it? Serine. Yes, by mistake, this car belong to serine. Anticodon to serine. Attached to valine. Valine is a thief. Valine came and took the car of serine. Now this valine attached with serine transport RNA was going. Now this amino acid transport RNA synthetase took 2 ATP, 2 energy, money. So he will keep in mind that if it is taken by valine, then who will pay? Yes, that's why this amino acid transport RNA synthetase has proofreading. What does it mean? Means it check. Again, I repeat. I say activation of amino acid. What is activation of amino acid? Binding of amino acid to transport RNA is called activation of amino acid. Binding of amino acid to transport RNA is called activation of amino acid. We simply say that this transport RNA is for serine, this transport RNA is for serine, so serine will bind to it, and in this binding, it two ATPs are used. Two ATPs are used. This activation of amino acid ultimately amino acid transport RNA is synthesized. Just like I showed you in this that was yes I showed you that look at this. I will tell you why anticodone I have discussed. Okay, for the time being, look at the screen. This is transport RNA for. Why I'm taking a little bit extra time in this? Because recently they have asked question on this and they have asked deep concept. Look at this. I say this is transport RNA for serine. How I will recognize that this is transport RNA for serine? I recognize this transport RNA of serine because it is having anticodone UGA. We always read from which direction? 5 to 3 direction. Okay. We found that this serine, this transport RNA, this transport RNA bind to serine. This binding of serine to transport RNA require 2 ATP. And what is the name of enzyme? What is the name of enzyme? Amino acyl transport RNA. What is the name of enzyme? Amino acyl transport RNA synthetase and use 2 ATP. But they are mismatching. There may be some adverse condition due to any reason. This transport RNA code for serine, and by mistake it is joined with valine. Then what will happen? This amino acyl transport RNA synthetase, this amino acyl transport RNA synthetase has proofreading. Means amino acyl transport RNA synthetase wanted to join serine, due to any reason it is joined with valine, alanine, whatever may be. This transport RNA belong to serine but attach valine. Now this amino acyl transport RNA synthetase has proofreading. Has proofreading means it can cut this valine. Suppose this is valine, it will cut this valine and will join serine again. So this is called proofreading. So question is asked, in protein synthesis, proofreading occur at which level? The name is amino acyl transport RNA synthetase has proofreading activity. Clear? Now what happened? Suppose this transport RNA, this transport RNA is for serine, and by mistake it is attached with. We found that this transport RNA by mistake attached with valine. Amino acyl transport RNA will do proofreading. It will remove this valine and will attach serine. But suppose this amino acyl transport RNA synthetase failed to correct it. Then what will happen? This transport RNA having valine, this transport RNA having valine will go and will bind to this place, and in place of serine, which amino acid would go to join the protein? Valine. Please do reply. Have you understood this? Now, frequently asked question has been asked previously in your PGI. Favorite question was there. Not a new question. It you can find these question in your Lippincott itself, Harper itself. So you can't skip these questions. Yes, again I repeat. How was question asked? Suppose protein synthesis is going on. In protein synthesis, transport RNA for serine is attached with valine. What would be the future of this transport RNA having valine? Okay, what would be the future of this transport RNA valine? Option A, it would be tried by amino acid transport RNA synthetase to correct it. This is correct option. Very good. Yes, if they give that this serine attached with valine, what would be the next step? This amino acid transport RNA synthetase will correct it. Will correct it. Next question. If transport RNA attached of serine attached with valine and amino acid transport RNA synthetase failed. Again, I repeat, amino acid transport RNA synthetase failed to do proofreading. Then, what would be the fate of this amino acid transport RNA having valine? What would be the fate? Now, this transport RNA having valine, this transport RNA having valine, yes, valine will not be discarded. Yes, option A. Yes, what would be the fate of this? Option A. Transport RNA will be discarded. Option B. This abnormal first option, abnormal transport RNA valine would be discarded. Option B. It will take part in protein synthesis, but in place of serine, valine would be in protein. Option C. It will take part in protein synthesis, but in place of valine, there would be serine in protein. What is the answer? Please do reply. It will take part in protein synthesis and in place of serine, who will join? Yes, very good. B. Yes, very good. It will not discard because codon recognize anticodon. So whatever sitting over there, we are not concerned. This will take part in protein synthesis. In place of serine, there would be valine. Very good. So if you could understand this, definitely it's a very good important question. Clear? Yes. Now we start protein synthesis. This is really interesting and important topic because till now we have seen protein synthesis, remembering protein synthesis without any concept. Now we will have visual memory. I will try to simplify it with the help of animation. Yes, protein synthesis, three steps are there. One is initiation, elongation, and termination. Initiation, elongation, and termination. First step is initiation. Actually, no dear. Codon for serine and valine are same, are different. Codon for serine and valine are different, but I told you that codon recognize anticodon. Codon recognize anticodon. Whosoever is sitting over there, we are not concerned. Yes, means the interaction is occurring at this level. If codon is for serine, anticodon of serine will bind to it. Transport RNA having anticodon will bind to it. If this transport RNA is having serine, valine, methionine, whatever amino acid, this is not concerned. Protein synthesis is not concerned with that. Whatever codon is there, anticodon means transport RNA having anticodon will come and bind to it. We are not concerned what is attached at 3. Initiation, elongation, and termination. Three steps in protein synthesis. First step is initiation of protein synthesis. What is initiation? Initiation means formation of initiation complex. Formation of, yes, formation of initiation complex. Yes, we found that initiation. In initiation, what are the steps? Yes, even if you don't remember, it's okay, but you must have an idea that what is initiation complex. Yes, look at the screen. This is messenger RNA. This is ribosome. Sorry, this is ribosome. You all know. First of all, ribosome. I am starting protein synthesis. In protein synthesis, initiation is the first step. Although few persons say that activation of amino acids should be considered the part of initiation protein synthesis, but we said no. Activation of amino acid will not be part of activation of amino acid is not a part of protein synthesis. Protein synthesis include only three steps: initiation, elongation, and termination. So don't get confused. No such question. Simply they ask, yes, important points you will remember simply. First is initiation complex. How it is formed? How it is formed? This is ribosome. First of all, these two parts of ribosomes are separated. After this separation, then messenger RNA and transport RNA having methionine comes. Yes. So you know this is transport RNA having methionine. AUG code for methionine. Question is asked AUG code for methionine. So you can write AUG code for methionine. AUG is codon for methionine, and AUG is also called initiation codon. Sorry, they are also called initiation initiation codon. Okay, we found that this methionine transport RNA having methionine will come and bind to this, and then this ribosomal unit will bind to this. You must have read this in pharma. Yes, I'm not going to discuss pharma in detail, but trust me, after this, revise your pharma antibiotics which are affecting this protein synthesis. It would be very easier for you to remember the classification. Yes, this is initiation complex. This is initiation complex. In this initiation complex, you will question is asked. Initiation complex is made up of messenger RNA, transport RNA having methionine, ribosome. Messenger RNA, transport RNA having methionine, and ribosome. Okay, now means first amino acid are always and always methionine. First amino acid is always and always methionine. Okay, now so this is initiation complex. I say you made initiation complex. In this initiation complex, yes, this is messenger RNA, and this is the initiation codon AUG. This sequence is called Shine-Dalgarno sequence. Shine-Dalgarno sequence. What is Shine-Dalgarno sequence? That is UAGG AGG. What is the significance of this Shine-Dalgarno sequence? I just told you that AUG is the initiation codon. And we found that suppose this messenger RNA, there are two AUG. Both AUG code for methionine. Suppose what happened? This protein synthesis is going on. Initial complex is going to be framed. Yes, we want to make initial complex. When I was about to make initial complex, I found that this messenger RNA come. Now you can see this is AUG, this is AUG. This is transport RNA having methionine. This transport RNA having methionine can bind to this side or this side. Protein synthesis should start from this side or this side, which should both are AUG, which AUG should be initiation codon. So we found that in messenger RNA near to initiation codon, suppose this AUG, yes, I told you that this bind to this side means this AUG. We found that near this AUG, we found that near this AUG there is a codon sequence, nucleotide sequence, and Dalgarno sequence. Means the starting point is that AUG near to which there is a Chantal-Carnot sequence. So we say that Chantal-Carnot sequence determine initiation codon. So Chantal-Carnot sequence is near AUG. Chantal-Carnot sequence is near AUG. So this is Chantal-Carnot sequence, and this is an initiation complex. Okay. Now, now, yes. Second step is elongation. Before going to elongation, I would tell you that in initiation complex, we found that in initiation complex, look at this. This is initiation complex. This is called P site. This is called P site. P site. P for polypeptide site. And this next codon is called A site. I can highlight like this. This is P site and this is A site. So this is initiation complex. In initiation complex, I found that this is Chantal-Carnot sequence. Near to Chantal-Carnot sequence, there is initiation codon AUG. On this, methionine transport RNA is attached, and the next codon is triple U. Triple U code for, yes, triple U code for phenylalanine, and this phenylalanine will come and will bind here. Phenylalanine transport RNA having phenylalanine will come and bind here. Yes. Now this means now onwards. Now I start elongation. In elongation, first step is binding of transport RNA, binding of transport RNA, binding of transport RNA to A site. Yes, shared diagonal sequence is present in all prokaryotic messenger RNA. Okay. Yes. Transport RNA to A site. Binding of transport RNA to A site. One step in elongation. So in first step protein synthesis, I discussed three steps: initiation, elongation, and termination. First step was initiation. I told you formation of initiation complex. Second step, I am telling you elongation. In elongation, again three subdivided into three steps. First is binding of transport RNA to acceptor site. Yes, this transport RNA will bind to this acceptor site, and binding of this require 1 GTP. 1 GTP. Now P site is having methionine, A site is having phenylalanine. Now the next enzyme which come into action, we found that in ribosome there was a RNA, ribosomal RNA, was having enzymatic activity. Enzymatic activity. The name of enzymatic activity is peptidyl transferase. Peptidyl transferase. So peptidyl transferase is the enzymatic activity. Peptidyl transferase is the enzymatic activity which is present in, yes, peptidyl transferase. Peptidyl transferase is the enzymatic activity which is present, yes, in RNA, and any enzymatic activity present in RNA is called ribozyme. So I represent, look at the screen, this is peptidyl transferase, this is peptidyl transferase. What is the function of this peptidyl transferase? Peptidyl transferase transfer, look at the screen, transfer this methionine, transfer this methionine to phenylalanine. This is peptidyl transferase. Now, and in this transfer, question is asked, peptidyl transferase transfer amino acid from P site to A site, from P site to P site to A site. Okay. Now, a question, I will ask you a question in a simpler way. Otherwise, you will find a difficult question. Protein synthesis is going on. Protein synthesis is going on. We are doing an experiment. We added antibiotic, and this antibiotic stopped protein synthesis. And when protein synthesis stopped, yes, during in the presence of antibiotic, we found that there is formation of dipeptide. Means lot of dipeptide was were there. Means, again I repeat in a simpler way, in a protein synthesis going on in the presence of antibiotic, and the resulting compound is dipeptide. Resulting protein is dipeptide. So which step is inhibited? Option A, initiation is inhibited. Option A, initiation is inhibited. No. Option A, initiation is inhibited. No. If initiation is inhibited, no formation of dipeptide, mono single amino acid would remain. Option B, binding of transport RNA to A site is inhibited. No, if binding of transport RNA to A site is inhibited, it is not, yes, it is not going to give rise to dipeptide. C. Very good, you have given the correct answer, but they are what they want you to be more precise. Peptidyl transferase is inhibited. Yes, if peptidyl transferase is inhibited, yes, if peptidyl transferase is inhibited, there can't be formation of dipeptide. There can't be formation of dipeptide. So peptidyl transferase. After peptidyl transferase, the next step is transloc. No, peptidyl transferase not inhibited. Yes, peptidyl transferase is inhibited. No, this is not answer. If peptidyl transferase is inhibited, then there would be no dipeptide, only monopeptide. Next step to this is inhibited. What is the next step? Now look at the screen. Peptidyl transferase transfer this amino acid from P site to A site. Now, when you will move that, yes, what happens after peptidyl transferase? There is a step. This is called translocation. Means this ribosome slide over messenger RNA. This ribosome slide over messenger RNA. So this is translocation. Yes, in elongation, third part is, yes, translocation. Yes. So again I write here. In after initiation, I write second step is elongation. Elongation has three part. First is binding of transport RNA to A site. Second is peptidyl transferase. Third is translocation. Okay, so translocation is the third component of elongation, and now you can easily see that what happened. Yes, yes, yes. This I will give you answer, dear. Okay, so this is now I start again. Initiation complex is formed. What is initiation complex? Yes, what is initiation complex? Look at the screen. This is initiation complex. Means P site, A site, transport RNA attached with P site, messenger RNA is there, ribosome is there. This is initiation complex. Now I start elongation. In elongation, first step is binding of transport RNA to acceptor site. This Yes, don't worry, I will revise it again. I know this is very complicated. Sorry for giving question in the middle, but if you will remember like this, it would be helpful in your exam. Okay, so we found that this is initiation complex. Now I start elongation. In elongation, first step is binding of transport RNA to acceptor site, and it require one GTP. It require one GTP. Then the next enzyme come into action, that is peptidyl transferase. The question is asked: peptidyl transferase transfer a amino acid or peptide from P site to A site. Then the next step is translocation. Yes means this ribosome slide over messenger RNA. Slide over messenger RNA. This is translocation. Suppose this translocation is inhibited. Suppose this translocation is inhibited, then further synthesis will not take place, and this dipeptide will be released. This dipeptide will be released. If translocation is inhibited, then this step would not occur, translocation would not occur, and this dipeptide would be released. So if dipeptide is released means the reaction up to peptidyl transferase has occurred. Got the point? So if I can accept that you are a little bit confused, now what I do, I again repeat, I leave it. For the time being, I leave it. For the time being, I leave it. Okay, I simply say that this is initial complex. Now elongation, first step is binding of transport RNA to acceptor site. It requires one GTP. Second is peptidyl transferase. Peptidyl transferase is ribozyme. It transfer, yes, methionine from P site to A site. Dipeptide is formed. Now translocation occur. Translocation is movement of yes, movement of ribosome over messenger RNA. This is translocation. I will repeat the question again. Don't worry. For the time being, yes. This. What was the question? Suppose you are doing protein synthesis. You added antibiotic. Protein synthesis is going on, and dipeptide is formed. If dipeptide is formed, it means protein synthesis has occurred till now. If before that any step is inhibited, if peptidyl transferase is inhibited, there would be no peptide dipeptide. If this binding of transport RNA to acceptor site is inhibited, no dipeptide. If initial complex is not formed, no dipeptide. Means if dipeptide is formed in in an experiment, it means that step up to peptidyl transferase have occurred. Now the next step is translocation. Next step is translocation. So translocation is inhibited. This step is inhibited. That's why the resulting peptide is dipeptide. Understood? Please do reply till now. Have you understood this? Now, next is what happened next. After that, yes, this become P site and this become A site. The next codon become A site. Next codon become ribosome move only one codon. Ribosome move only one codon. Yes, move one codon. Don't worry, I will make you understand. Don't worry. I will summarize in the last. It will be clear to you. Don't worry, Sneha. Yes, A site. A site becomes free. A site becomes free. I know this is a difficult part. Yes, either I can tell you in a simpler way. Yes, but that would not be able. You will not be able to solve question in the exam. That's why I'm giving a little bit more stress and more in a difficult way. Okay. Now A site. Now this A site AUG code for methionine. Then the transport RNA having methionine will come and again bind to A site. Binding of this A site require 1 GTP. 1 GTP. Next, what will happen? Binding after binding. Yes, after binding. Yes, sorry. After binding, peptidyl transferase would act. Peptidyl transferase will transfer these two amino acid to methionine. Yes, and then translocation occur. Clear? Translocation occur. So elongation include three steps. Elongation means transport RNA to A site, just like it binds transport RNA to A site. Then peptidyl transferase transfer from P site to A site. Then translocation occur and AUG remains outside. And next codon become P site. This codon become P site, and the next codon become A site. Now the AUG code for methionine. That's why transport RNA having methionine will come and bind to it. Now after that, what would happen? That translocation would occur again. Sorry, before that, will peptidyl transferase will transfer these two amino acid to this methionine, and then translocation will occur. Okay, I know I have made it little bit difficult by introducing the question, but don't worry. Now look at what is happening. This is initiation complex. In this initiation complex, this is P site and A site. Now trans A site has triple U. A site is having triple U. That's the reason that transport RNA having phenylalanine, triple U code for phenylalanine. That's why transport RNA having phenylalanine will come and bind to acceptor site. Now peptidyl transferase will transfer methionine to phenylalanine and dipeptide will be formed. After that, translocation would occur and AUG will remain outside. This codon will become P site. Next codon will become A site. Now, this transport RNA having methionine, because AUG code for methionine will come and bind to this site. Again, require 1 GTP. Again, peptidyl transferase will come and will transfer 2 amino acid to methionine to make peptide bond. Yes, and then again translocation occur and this codon comes outside. This become P site and the next codon become A site. Till now, is it clear? So this is elongation. Elongation, yes. Whenever you push anything, you require energy. You push anything, you require energy, and we found that translocation require 1 GTP. Translocation require 1 GTP. 1 GTP here, 1 GTP here. Okay, very good. 1 GTP here, 1 GTP. Now the last step is termination. Last step is termination. Okay, yes, very good. Now look at this. This is P site. This is P site. This codon becomes A site. Now I am starting termination. Question is asked, when protein is terminated, synthesis is terminated. First question, when stop codon comes at acceptor site, UAG stop codon came at acceptor site. When stop codon comes at acceptor site, this releasing factor will recognize this stop codon. When stop codon comes at acceptor site, I write here releasing factor recognize stop codon and activate peptidyl transferase. First question, can you tell me answer? Newly synthesized polypeptide chain is released by which enzyme? Newly synthesized polypeptide chain or protein is released by which enzyme? Newly, yes, releasing factor recognize stop codon and activate peptidyl transferase. Peptidyl transferase cut polypeptide chain from, yes. Now peptidyl transferase cut the polypeptide chain from, yes. This releasing factor recognize. Look at the screen. Releasing factor recognize stop codon and activate peptidyl transferase. Now peptidyl transferase go and release polypeptide chain from P side. From which side? P side. Clear? It is misnomer. Don't get confused by name. Releasing factor don't release. Releasing factor recognize stop codon. When stop codon come and acceptor site recognized by releasing factor. Releasing factor, yes, releasing factor activate peptidyl transferase and polypeptide chain is released from P side with the help of which peptidyl? Yes, termination. Yes, in this releasing factor recognize stop codon activate peptidyl transferase and newly synthesized polypeptide chain released by peptidyl transferase. This is released. This releasing factor is not ribozyme. Releasing factor is a, yes, releasing factor is a protein having enzymatic activity. Okay, clear? So this is protein synthesis. Now just have a look of which we have done. Yes, this is P site. Next is A site. This is initiation complex. Now we start elongation. In elongation, yes, first step is transport RNA bind to acceptor site. Next enzyme peptidyl transferase. Peptidyl transferase transfer methionine to first amino acid to phenylalanine from P2A site. After that after that translocation occur, means ribosome slide over this messenger RNA and one codon comes outside. This codon comes outside, AUG. Now the next codon become acceptor site. Again next transport RNA come and bind to acceptor site. Again peptidyl transferase transfer these amino acid to methionine. In this way, every time one amino acid is joined. When after so many amino acid joining so many amino acid in this protein, only 3 amino acids are there. Stop codon comes UAG. Then releasing factor will recognize stop codon and will activate peptidyl transferase, and peptidyl transferase will release polypeptide chain from P site. This is protein synthesis. And the question was that if a patient, if you are doing experiment and you added antibiotic is inhibiting protein synthesis. Up to this step, up to this step means if peptidyl transferase is wrong, is not functioning, you can't make peptidyl trans, you can't make dipeptide. If transport RNA binding to a site is defective, you can't make dipeptide. So if dipeptide is formed in protein synthesis, definitely the step inhibited is translocation. Got the point? So this is protein synthesis. I tried to give you in a difficult way. Yes, but you will find so many questions in MLE pattern. Not only in MLE pattern, these question has been asked in PGI previously, and these are common prevalent questions of protein synthesis inhibition in your Lipin code Harper. So they can pick up any question and they can ask you. And if you remember this visual memory of these events, it would be easier for you to understand.